Hello, good morning my dear student. Here myself, Dr. B.N. Thorat, Assistant Professor, Department of Animal Husbandry at Dairy Science. We have conducted today's theory session. In that theory session, we have taught the first topic, that is history and concept of animal breeding. On this topic, always asking 4 mark questions in your final theory examination as well as in your midterm examination. So going to breeding, going to concept as well as brief history, we have required some brief information regarding this animal breeding. For the successful selective breeding, important aspect is the traits. Likewise, for example, milk production and coat color. Those animal which is having different genetic backgrounds, that in that animal selection is very possible. The direction of selection is defined by human, and they decide which animals are allowed to mate and produce member of next generation after generation. For the successful animal breeding, we can judge by looking at a shift in population average, then phenotypic form from one generation to another generation, so that animal breeding work at a population level but not automatically at individual levels. Success of animal breeding can be measured as the cumulative result of multiple generation of a selection. Then breeding decides are made with a future in our mind. So in that, first of all, we see the definition of animal breeding. So what is meant by animal breeding? So animal breeding means the application of principles of a population genetics by using the biometrical techniques for genetic improvement of livestock is known as animal breeding. So let's move on most important part, that is history of animal breeding. So in that history, this history of animal is start in the 18th century. In Europe, the origin of animal breeding lays in the United Kingdom. It was Sir Robert Backwell. So what is the work of Robert Backwell scientist? They have worked lots of in genetics as well as in animal genetics or animal breeding. He introduced the keeping accurate record of performance of animal so that objective of selection become possible or easy. Then another, he used inbreeding. Inbreeding means mating of related animals with similar traits of animal to fix certain characteristics of animals. They also introduced different type of progeny testing. The methods of evaluating performance of the first, that is small group of progeny, and use that the information to select and the best father of future progeny. He remembered for setting of pattern of model animal breeding as well as these all work considering they have promoted as the breed the best to the best. That's why the Robert Backwell has known as a father of animal breeding. All of you remember who is the father of animal breeding, always asking in objective type questions. So who is the father of animal breeding? That is Robert Backwell. Then Robert Backwell also developed the new Leicester sheep from the old Lincoln sheep breed. The new Leicester had good quality flesh as well as good quality fat shoulder that was popular at that time. Backwell also noticed the Leghorn cattle were growing well and used less feed compared to the other cattle. So he developed that feature in order to grow more for the meat efficiency. It is amazing he did not without knowing anything about the genetics. So in that history of animal breeding, some Indian and outside scientists, they have also worked. So which is the work of Devicky. They relieve the fact that male and female parents contribute equally to the heredity of offspring from one generation to another generation. Then which is the contribution of Robert Hooke. They apply the term cell in animal genetics. Then again Robert Backwell, they have first to start the breeding and establish breeds. He is setting pattern of modern animal breeding. That's why by considering their work, they are the founder of animal breeding or father of animal breeding. Then which is the work or contribution of Spallanzani? They have used artificial insemination successfully to inseminate in spinach beach. Then another scientist, they say that they first account the terminology that is mitosis. So what is meant by mitosis? Likewise, then Mendel, all of you know that they are the father of genetics. They have worked lots of work in the genetics as well as in plant breeding. They are the Australian monk has called father of genetics because he worked first to uncover and record of the inheritance, segregation, independent assignment of a gene. Then Muller, H.J. Muller scientist, they have reported the artificial induction of mutation in animal by X-ray. So what is meant by mutation? Mutation means the sudden change in genetic makeup of an organism. Then Sampath Kumar, they have also worked the first attempted artificial insemination at Royal Palace Dairy Farm at Mysore. Then Lush scientists, they have also most important and always asking regarding your objective or in contribution of scientists. They have worked on the quantitative traits measurement, then progeny testing, then computerized data and management, then measure traits from birth to date, that means pedigree sheet. That's why they have called father of modern animal breeding. Then next scientist is a Wilmot. They have produced a clone of adult lamb and named as a Dolly. Then another scientist, that is William Batson. Batson had coined the term genetics. Then Johnson scientist, they have also worked on this coin the allele or genes, which is introduced the concept of genotype and phenotype. In that all of you are taught different terminology, what is meant by gene, genetics, allele, then phenotype, genotype. Likewise, what is meant by allele? Allele means alternative form of a gene. So gene is the structural and functional unit of a heredity. What is meant by genotype? That is genetic makeup of an organism, and phenotype, that is a external appearance of an organism. Then Charles Darwin, they have also published the book that is known as Origin of Species and Origin of Species as well as Theory of Evolution. Then Weismann, they have put forward the germplasm theory. Then another most important scientist is Batson and Punnett. They firstly discovered or observed linkage in a sweet pea. Then Strasbourg scientists who first discovered chromosome. Then Wilder, they have coined the term chromosome. Then Charles Darwin, in the year of 18, they have proved the natural selection, genotype by environment and interaction. Then in animal breeding in India, some specific scientists, they have worked in India on the animal breeding. In that, first upon the Sampath Kumar in the year of 1939, they have first attempted artificial insemination in cow at Mysore Royal Parks. Then Sukatme, they have statistically analyzed the data of goat breeding at Indian Council of Agriculture Research. Then Krishna, they have developed new sire indices. Then Gopalan studied the effect of linkage on quantitative traits. Another one, Sudarshan and his colleagues or their colleagues, they have developed corrected daughter average index. Then Narayan and his colleagues in the year of 1963, they have studied gene action and interaction with the multiple allele. Then Tomar, they have predicted what is the body weight of cow from the body measurement. Then Tomar SS scientists, they have developed different books regarding this animal breeding, and they have also developed methods of estimating heritability of threshold traits. Then Wilmot scientists, the cloned sheep Dolly from the somatic cell of adult ewes. Then Johnson, they have worked on the pure line concept, then coined the words gene. Genes mean that is structural and functional unit of a heredity. Then gene, single unit of a heredity. Then another Nielsen-Ille, they have discovered the nature of heredity. Then Irwin and Carl Correns, all genes do not segregate independently, but some tends to have together in the next generation. Then Wilson, they have the pioneer in cytogenesis, they have discovered the relationship of the chromosome of the cell to the heredity. Then another scientist, they have worked that genes are composed of a specific type of a nucleic acid, that is, all of you know that DNA. DNA means deoxyribose nucleic acid, not a protein. Then another, Tatum and Budel, that is put one gene, one enzyme for the hypothesis. Then Watson and Crick, these scientists all of you know because they have worked on the structure of DNA. These are the nucleic acid in the 1953. Then Monon finally provide genetic evidence for the method of gene regulation in bacteria. Then another, Lamarck. He is best known for use and disuse as well as inheritance of acquired characters. Then Weismann, they have worked on the multicellular organism passes germplasm, which is preserved by the repeated division. Germ cell came from the germplasm, other cell which is came from the somatoplasm. Then environmental changes that occurs only in the somatoplasm and not in a germplasm. Germplasm transmit heredity from parent to their offspring or generation after generation. Then acquired characters could not be therefore be inherited. Germplasm is only responsible for formation of new cells. Then Thomas Morgan, they have coined the term linkage, in which genes located on the same chromosome which are traveled together in process of inheritance, resulting in the appearance of parental types in greater number than recombination. The first genetist to receive Nobel Prize in Biology and Medicine in the year of 1933. Then there are different important objectives of animal breeding, which are that on this objective one question is asking in your midterm as well as final theory examination. Write down the objective of animal breeding. So there are first there are number of objectives. To first one, to improve the breed and herd of animals, to select the animal of a desired types, then to improve growth rate as well as performance of a non-descriptive breeds of a local breed of a animal, then to develop early maturity in animals, to increase the quantity of product, that means milk production, meat production, egg production, wool production, etc. Then again, to improve the quality of products, in that we have required to improve fat percentage, then flesh quality, egg size, egg production, etc. To improve the productive life of animals. Then again, to improve the adaptation of animals in different environmental conditions, to develop immunity against different type of bacterial, viral, fungal diseases as well as contagious and non-contagious diseases, to reduce the immature mortality rate in animals, to improve body weight gain in animals, and last one, to increase the daily income of dairy farmers. Then which are the most important concept of animal breeding? So in that concept, breeding of livestock for environmental adaptation, use of new techniques in animal, then improvement of quantity and quality of products of animal. Again, breeding for disease resistance, then improvement in selection of animals, then again increase the income of dairymen, as well as the latest technique, that is in latest techniques we have to use cloning of animals and production of transgenic animals. Then also we have to use advanced technique like artificial insemination, multiple ovulation, as well as embryo transfer technology, and also use techniques like the inbreeding, then outbreeding with the knowledge of genetics. These are the different basic concepts which is used in livestock breeding. So thank you all of you for your patience. If anybody having any queries, so ask me, I will be explain again. Thank you.